architectures. So some of the challenges that are faced in terms of implementing these genome LLMs are the pre-training tasks, the computational cost, and their lack of interpretability. And I'll finish off by talking about the future of genome LLMs as I see them, which includes multi-species training and unsupervised pre-training. So to begin, what are genome LLMs? Well, genome LLMs are essentially a class of models that are trained on a specific kind of data, specifically genomic sequence tokens. And these can come from sequential genome data, like whole genomes or specific genomic regions. And they can actually be created from non-sequential genome data as well. So this would include single-cell transcriptomes. And essentially, both of these data sources can be tokenized, and a genome LLM pre-trains on this data. Later, it can be fine-tuned for a variety of downstream tasks. And these downstream tasks can include functional region identification, disease single nucleotide polymorphism predictions, and gene expression level predictions. So as you guys all probably know, genome LLMs are now being widely adopted in genomics. These models include DNA-BERT, GeneLM, SCGPT, HyenaDNA, GeneFormer, Nucleotide Transformer. More and more we see these genome LLMs are coming out and being published. So to begin, I'm going to talk a little bit about the architecture of kind of the main model that is used for these genome LLMs, and that is the transformer. And the transformer is particularly interesting, obviously, because of the success of the GPT models and things that we're very familiar with. Kind of what brought AI to the attention of most people was the GPT and the transformer. So essentially, the transformer is a sequence-to-sequence architecture, and it's composed of many stacked layers. So the transformer isn't a specific layer type. It's actually kind of like a collection of stacked layers that includes like layer normalizations, skip connections, and all sorts. So usually what goes into the model is some kind of sequence input. In this case, I'm demonstrating it could be a DNA sequence that's taken as input. And then it goes through the model to produce input encodings as well as output probabilities. And there's two main components of the transformer architecture that was initially proposed. There's an encoder and a decoder. And kind of what separates the encoder and the decoder are different combinations of these stacked layers within the module. And they also have kind of a unique layer organization and purpose. So the encoder is used to produce embeddings or input encodings, and the decoder is used to help produce these output probabilities that can be used to generate sequences. So I'm going to be focusing more on the encoder-only transformers, and that's simply because they're more common in the case of genomic applications. So to begin, I will talk about the attention mechanism, and that is kind of one of the main aspects of the transformer model that is attributed to their success. I personally want to talk about the attention mechanism because it is not very well understood, including by me for a long time, and I think it's very interesting in understanding the success of these models. So to begin, the way that we can think about attention is kind of calculating a relevance of every position in an input sequence to every other position. So you can imagine in the context of DNA sequence data, we may want the relevance of a position in a promoter sequence to be very strongly related to sequences in the relevant gene that the promoter is associated with. You would expect there to be strong relevance between those two subsequences. But you would expect the promoter maybe to pay less attention to a non-coding region of DNA that has no regulatory effect. So what is the attention mechanism? The attention mechanism is kind of summarized by this very simple equation that has been seen probably by a lot of you. It's essentially a softmax over the dot product of the queries and keys divided by a scaling factor, and then multiplied again by a values matrix. And a lot of the time people leave the explanation of attention at that, but it doesn't really make a lot of sense unless you know what these queries and keys and values are. So keys, queries, and values are linear transformations of the input sequence. We take a DNA sequence and we tokenize it in some way there's a variety of ways to tokenize this data. And then these tokenized inputs are linearly transformed or projected into queries, keys, and values. And so these keys, queries, and values are actually learned by the model to be different. The model learns parameters in order to determine what the queries, keys, and values are. So every single one of these matrices of the query matrix, the key matrix, and the value matrix, a single vector, so for example, a query vector is associated with a single token. And essentially what we're doing when we're creating these queries, keys, and values is we're computing a dot product between input embeddings and the three different matrices, the three different weight matrices that transform the input embeddings into these queries, keys, and values. And you can think about this as taking the input space and learning a meaningful embedding of the input space. So each of these matrices learn an embedding of the input space. And ideally, that embedding that is learned is, captures some of the semantic meaning of the tokenized sequence. So then we take the queries and the keys and we compute a dot product. And that dot product essentially measures for every token how related the two tokens are. And then we do a softmax. And that is the result of the dot. The softmax is run over the result of the dot product between the queries and keys. And the resulting matrix that is produced is the attention matrix. And this is important because this attention matrix is later used to interpret these models. So at a high level again, the attention matrix, what is it? So we learn these meaningful embeddings of queries, keys, and values, and these are representations of the input sequence. The first thing we do is we dot product the queries and the keys, and that is essentially to kind of determine the relatedness of every token to every other token, as the dot product is higher for tokens that are closer in this embedding space, this meaningful embedding space, and smaller for things that are less related. The reason that we do a scaling factor before the softmax is essentially for numerical stability. And then we run the softmax over the entire query and key dot product. And the reason we do that is to normalize the dot product scores between 0 and 1. And this is important because when we multiply by the value matrix, essentially what this does is because we've softmaxed everything between 0 and 1, this means all of the tokens that have not very relevant, that are not very relevant to one another are essentially going to be sent to 0, and everything else will retain its value. So those meaningful embeddings that were learned by the value matrix are then added to the original embeddings if they're relevant to that token. So I just talked a lot about attention. Many of these transformer models actually don't use attention broadly. They use something called self-attention, and that's what I was describing. So self-attention is always within the input sequence that's inputted to the encoder. So all of the tokens relative to each other, as I was talking about. Now, what's actually used in these models is multi-headed self-attention, which is very similar to attention. You just have more attention heads. And I actually find understanding multi-head self-attention was initially very confusing for me, but breaking it down in this way, I hope that it can be understandable. So it's essentially the same calculation for the attention matrix, except now instead of just learning one embedding of queries, keys, and values, we now have multiple different attention heads, and we're going to learn embeddings for queries, keys, and values in every single head. So if you imagine the creation of the query, key, and value matrix as putting the input tokens into a meaningful embedding space that captures some kind of semantic meaning. So you would hope that a token, and once it's been projected into the embedding space where the keys and queries are, a token that represents this promoter sequence is actually very close to the token that represents part of the gene sequence that promoter is related to, such that when they do the dot product, it's very high. So we can potentially learn many different meaningful embeddings of the input sequence if we have multiple different heads, since we're learning multiple queries, keys, and value matrices. Essentially, we just repeat the attention matrix calculation for every single one of the heads. And then this allows us to learn many different meanings. So this is multi-head self-attention, which is a very important aspect of the encoder. And I kind of haven't really been talking about the decoder, as I said earlier, because it's not as relevant for genomic transformers, but the decoder has both multi-head self-attention and multi-head attention, or it's sometimes called cross-attention, between the encoder and decoder. All right, so one of the other features of transformers that makes them so interesting and has been attributed to their success is that they are capable of pre-training and fine-tuning. They are not the only model architecture capable of pre-training and fine-tuning, but they are maybe one of the most prominent. And the reason this is so important is because, a lot of the time, or sometimes we may not have enough task-specific data to train a deep learning model. So we have this very large model that has very many parameters, and we don't have enough data points to train it. However, if you can use pre-training and fine-tuning, you can potentially train on data that doesn't have labels in an unsupervised way. So you can take a unlabeled large data set that's not task-specific. For example, we could just take the entire human genome in the case of large language models for genomics, and we could train the transformer model on this large unlabeled data set, and then adjust the model again, train it again on a smaller task-specific data set to achieve a specific, like a task-specific performance. So like in the context of genomics, you could imagine training on the whole genome, pre-training on the whole genome, and then fine-tuning on a specific task, for example, classifying enhancers from non-enhancer regions. And the goal of this kind of pre-training and fine-tuning approach is that you can leverage general insights from pre-training and use those to help inform predictions and fine-tuning. So what's the difference between a transformer model that's applied to genomics and an actual large language model? Well, many of the transformer models that are applied to genomic data are actually hybrid transformer models that are trained in a supervised manner on label data. So they don't undergo this pre-training and fine-tuning. These hybrid transformers can have other layers involved other than specifically the ones that define the transformer modules. They can include convolutional layers or recurrent neural network layers. In this case, I have a schematic of a genomic transformer model that's specifically a hybrid model. So it takes as input a sequence, and these sequences have specific labels. We can then tokenize the input sequence, or in this case, I'm showing a one-hot encoding of the input sequence. And then the model does some initial convolutions on the one-hot encoding before the information summarized by the convolutions is sent to the transformer. And these hybrid transformer models output information like qualitative track outputs, like Informer is a very common one to think about, or even like quantitative track outputs like chip peak intensity. So some examples of hybrid transformer models are Informer, as I already mentioned, Satori, Borzoi, and C-origami. To kind of dive in a little bit more to what these hybrid transformers look like, Informer takes as input a 200,000 base pair sequence and goes through a series of convolutions and then these transformer layers, and finally some organism-specific prediction heads that output these genomic tracks that include like gene expression, DNA accessibility, histone modification data, and so on. I believe there's actually over 5,000 tracks that Informer predicts for humans for a given sequence. All right, so moving on to the large language models. These large language models are different from the hybrid transformers because they're actually pre-trained on genomic tokens and then later fine-tuned for downstream tasks. So you take a DNA sequence and you tokenize it. You then use the multi-headed attention that I discussed. So you take these tokenized inputs, you project them into meaningful embedding spaces or into queries, keys, and values. And then you perform the attention calculation across all of these heads and take the information at the end. And you use this to kind of make a classification decision. Usually, for example, these can be fine-tuned to identify promoter versus enhancer sequences. So some examples of these transformer LLMs include DNA-BERT, nucleotide transformer, DNA-BERT2, and GinaLM. And to dive a little bit more into what these large language models kind of look like. So the nucleotide transformer is actually a series of models and it can take up to 6 kbp as input, and then it outputs a variety of different predictions based on what the model is fine-tuned to do. So the model is pre-trained broadly on the whole genome or different data sets of genomes, and based on which model it is. And then it can be fine-tuned to predict information like chromatin accessibility, transcription factor binding sites. It can predict enhancers and promoters, epigenetic marks, and so on. So there's a variety of different tasks that this model can be fine-tuned specifically to perform on. Another example is DNABERT2, which can take in kind of a broad range of sequence lengths based on the specific tokenization scheme that the model uses. And it can be fine-tuned to do a variety of different tasks in humans, mice, yeast, and viruses. And actually, one notable thing about the DNABERT2 paper is that along with proposing a model, they also propose a series of data sets in order to kind of create like benchmarks to evaluate these large language models, because currently a lot of these large language models are evaluated on different data sets and different tasks, and it's less straightforward to compare their performance. All right, and then there is the SCGPT model, and this model is kind of different from the other models that I've talked about because it actually takes as input essentially a non-sequential genome information in the form of single-cell RNA-seq data, but it is tokenized into an input the transformer can accept. And as output, it can be fine-tuned to do clustering, batch correction, cell type annotation, multi-omic integration, perturbation predictions, and gene network inferences. So I'm talking a lot about the transformer and how great it is. It has this amazing attention calculation. It can do pre-training and fine-tuning. One of the drawbacks of the attention mechanism is that it is bounded by a quadratic cost in sequence length. And that is essentially because attention is this computation of relevance of every position to each other position. So as the input sequence expands, the attention calculation goes up quadratically. And this is kind of one of the limitations now of these transformer models, is that increasing their context size is very computationally expensive. So as a result, you may often hear about these alternatives to the attention mechanism being proposed. So that's like essentially you have the exact same transformer architecture. So the series of stacked layers we're familiar with that define the encoder and the decoder. And the only thing you do is you just replace the attention calculation with a different version of attention. So there are a few that have been proposed, but one of them is this local attention, where instead of calculating attention across the entire sequence window or context length, you subset and only calculate attention in smaller areas. And one of the very popular transformer alternative architectures that has been proposed is the Hyena model. And essentially the Hyena model proposes the Hyena operator in place of the attention mechanism. And what the Hyena operator is it's an implicitly parameterized long convolution and data controlled gating. So essentially what this means is, instead of running attention across the whole sequence, what they do is they run the Hyena operator across the whole sequence. And what that is essentially a convolution equal to the sequence length. And the way that they avoid the quadratic cost of having a convolution equal to the sequence length is by doing a fast Fourier transform. All right. So I'm talking about LLMs. I'm going to get into some of the challenges that these models face. And one of the challenges that they have are these pre-training tasks that they undergo, or these pretext tasks. So I said at the start that these genome language models are defined by pre-training on genomic tokens and later being fine-tuned to downstream tasks. But what is this pre-training that these models undergo? So before I get into that, one of the things about LLMs that is part of what makes them so powerful is that they're these huge models. They have huge numbers of parameters, and they sometimes require billions of data points to train, in the case of the nucleotide transformer. So when I talk about transformers, I said kind of like one of the main things that is beneficial about the transformer is that it can be pre-trained and fine-tuned. Now, the thing is, once you move into the territory of having a large language model, it's not about it can be pre-trained and fine-tuned. As soon as you have a model with this many parameters, you are required almost to fine tune, to pre-train. You have to pre-train just because these models are so big that we don't have enough labeled data. So now every time there is a large language model for the genome, they're required to do some pre-training because there just isn't enough task-specific data for any task. So when I introduced the idea of pre-training and fine-tuning, I talked about how we can pre-train on some unlabeled large data set, for example, the human genome. And, we can achieve some kind of accuracy on this pre-training before fine-tuning on a smaller task-specific data set. And what I didn't really talk about was, what does the model train to do if it's unsupervised? The unlabeled data set has no labels, so the model can't learn to predict labels, and this task can't be measured. So what do we evaluate the model on? So in pre-training, we use kind of these two main pretext tasks to direct unsupervised learning. And both of these tasks are based on reconstruction. So they essentially try to hide some part of the inputted data and then have the model reconstruct it. And there's these two main tasks that I've been talking about. One of them is masked language modeling, where you mask out a specific token in a sequence, and the model kind of uses information from neighboring tokens preceding and afterwards in order to reconstruct the missing token. Now, autoregressive language modeling, or it's sometimes called next token prediction, is another pretext task that's used to pre-train these models. And in this case, it's again a reconstruction task. So some tokens are hidden, except the model can only use preceding information to fill in the missing tokens. So autoregressive language modeling only allows the model to look at what was before, and masked language modeling allows the model to look at surrounding context in order to fill in a masked token. So these tasks work well in NLP. We can clearly see with ChatGPT and the other GPT models that they have very strong performance. But the question is, how well do these tasks work in the context of genomics? So a recent investigation of the BERT model, so like DNA BERT, so a BERT model for genomics, essentially found that using the K-mer embeddings pre-trained on random data yields similar performance in downstream tasks compared with those on real K-mer embeddings. So essentially what that means is they kind of use this DNA BERT style model, and they pre-trained it on K-merized versions of the human genome, and then, evaluated it after fine-tuning and checked how it performed on a bunch of tasks. And then they also took this same model, instead of pre-training on K-merized version of the human genome, they essentially generated random sequences of nucleotides and then K-merized them and pre-trained the model on those random K-mers. And they found that after fine-tuning, the model performed pretty much the same. So essentially what that means is that this pre-training task where the model is supposed to be learning some underlying grammar of the human genome is actually not directing the learning of the model as much as we'd expect, because it performs just as well with random. So this other paper that recently came out by Tang and Ku evaluated the embeddings of these genome language models. So this is after pre-training and before fine-tuning. You can take the embeddings of these models, and what they did was they tested a variety of different models. They used the nucleotide transformer, generative pre-trained network, the hyena model, DNA BERT2, and then this is a supervised pre-trained model, and this is a model that they trained specifically for some tasks. And essentially what they did was they took the embeddings of these models and then used these embeddings to train a shallow convolutional neural network to do some kind of predictive tasks. And then they also just used a simple one-hot encoding and trained a shallow CNN to do these tasks, including alternative splicing or RNA elongation. And what you can see here is the performance of all of these models on these tasks is the performance of their embeddings being fed into a CNN model and then used to predict. And you can see the average Pearson correlation of these tasks. And essentially what you're seeing is that the one-hot encodings outperform the embeddings of these pre-trained models. So essentially the embeddings of these large language models for the genome are not providing additionally biologically relevant information for the CNN to learn, as opposed to simple one-hot encodings for these tasks. Another issue with LLMs is their compute. So what I mean by this is here we can see on the x-axis the parameter number in millions for these models. And on the y-axis, we have PFS days. And the way you can think about these petaflops days is this is a measure of compute which has been proposed by OpenAI, the creators of GPT. And they can kind of these PFS days are a way to compare how much compute these models take, which takes into account what architecture, which takes into account kind of like the architecture of the model, like the parameters, as well as like what specific hardware they were trained on. And this can be thought of similarly to kilowatt hours for energy. And what you can see here is that like many of these transformer models, even not just large language models like Informer, they take a lot of compute. Basically here, this line shows the equivalent of $5,000 worth of PFS days. And what you can see is that, these Hyena DNA Informer and nucleotide transformer takes over $5,000 worth of compute to train. That also means that if you want to work on these large language models and you're in an academic lab, it's very expensive, one, to pre-train these models, and two, to design and test and iterate on these. So the last challenge I'm going to be talking about is the interpretability of these models. Now, these are huge models with many parameters. And the question is, how do we interpret them? And the short answer is that we can't yet. So there have been a few proposed ways to kind of interpret these models, and this includes, using the attention mechanism itself. So for DNA-BERT, the original paper, what they did was they proposed kind of a visualization of attention landscapes. So what you can see here on the x-axis is like the length of a sequence, and they've labeled the positions with the distance in base pairs from the transcription start site. So this is where the transcription start site is, and this is, upstream from that, and this is downstream. And then on the y-axis on this end, we have like a variety of different sequences. So, like about 2,500 sequences here, and then, closer to 300 here. And over on the bottom here, we can see the average attention on these sequences as computed by the attention mechanism. And these are split into two different types of sequences. So this set of sequences doesn't have a Tata box. It's just like regular sequences. And then these sequences actually do have a Tata box just upstream from the transcription start site. And what they're showing here in these attention landscapes is that, for sequences that actually do have this Tata box, there is more attention being there's more attention being placed on this region of the sequence upstream from the TSS, as opposed to like the sequences that don't. So what they're trying to say is that the attention mechanism shows that the model has actually learned that Tata boxes are important to differentiate these sequences. Another way that we can try to understand these models is using their embeddings. So this is the SCGPT model, and they actually took the pre-trained embeddings from these models. So this is prior to fine-tuning. And they actually found that they could group functionally related genes and differentiate functionally distinct genes from these learned gene token embeddings. All right. So I said a lot about genome LLMs, and I pointed out that they have some serious limitations currently. They struggle to find biologically meaningful representations during pre-training. They're difficult to interpret, and they take a lot of compute. So, what is their future? So as I was talking about, like, why would we even worry about like interpreting these models if they don't actually perform that well in pre-training? And, why would we want to improve them even if they're not doing well? So one of the main reasons is scaling laws. And this is actually something that was proposed by the OpenAI team. And essentially what they found was that language modeling performance improves smoothly as they increase model size, data set size, and amount of compute used for training. So here on the x-axis of this model, you can see like the compute increasing, and they've shown this PF days, this measure of compute that we talked about. And they also have, data set size and parameters, and you can see test loss, which is equivalent to model performance. And as you can see, there's kind of like this decreasing relationship. So loss goes further and further down as compute increases, as we train on more and more data, and as we increase the size of these models. So this is like essentially the scaling law of language models, which have this distinct property that basically says, if we put more compute towards them, if we train them on more data and we increase their size, we'll just keep increasing and increasing performance, or dropping and dropping loss, and eventually, we'll reach some, like ideal performance metric. So language models have this unique property that makes them worthwhile to invest in. The other opportunity is that these genome language models can be trained to be generative. So discriminative models obviously do some kind of classification or do some kind of differentiation, and generative models produce new sequences in the case of sequence models, and in the case of DNA, they could produce new genomic sequences. And, part of that is really interesting because we could conditionally generate sequences based on specific experimental values that we have. And more importantly, in my belief, is that we can generate sequences, DNA sequences, that allow us to kind of train more models on generated sequences, because they're not private data. So essentially, a lot of these genome language models are currently constricted by the fact that for human genomes, they have to train on the reference genome, or they train on a very limited subset because genome data is private and identifying. So if we could train a model to generate this genomic data that matches the distribution of the human genome without having the drawback of revealing private information, it could potentially really help in training these models. And furthermore, not just for training these models, generated DNA sequences could be used for tasks beyond machine learning. And the last reason that I think these genome LLMs are so worthwhile in investing in is because of their unsupervised pre-training. And I talked about how they're not doing well in their unsupervised pre-training, but just because the models aren't currently leveraging the full benefits of pre-training, it doesn't mean they can't. And there's this, famous paper that came out, which is, the future is unsupervised, which is, the future of biology and of uncovering novel biology is in this unsupervised learning. And that's because, we don't impose our prior knowledge or, biases on training these models in order to discover something. We can just let the models learn themselves from the data, and that's very powerful once we get it right. All right, so what is the future of these models now that I have hopefully convinced you that they do have a future? I think that I personally think that there's huge potential in unsupervised multi-species pre-training regimes. So what I mean by that is some of these models only train on the human genome, but some of them train on the human genome, the mouse genome, and so on. So the goal in training on multiple genomes is to capture evolutionarily conserved data in the genome and to better model its underlying grammar. And the idea behind that is, evolution has already determined kind of what regions of the genome are important, because they're the ones that don't change a lot between species. And actually, the nucleotide transformer paper, they tested a variety of different models, and they tested them on different data sets. So they had multi-species data sets, 1,000 genome data sets, just the human reference. And what they found was actually that evolution informs representation. So models trained on genomes from different species actually performed better than models that were trained on just the human reference genome alone. And this was not just for human-specific tasks. These models trained on a variety of different genomes outperformed just the human genome trained models on human-specific tasks. I also think there's a future for these models in designing more meaningful pre-training tasks. And I think this could greatly remedy the LLM limitations. I think this is because, we're kind of using these pretext tasks, we're borrowing them from natural language processing, where there's very distinct words and structure to language, and it's less obvious in the genome what the equivalent of words would be. And this is kind of shown by the fact that maybe we could design a pre-training task that where training on a random k-mer versus true genome k-mer would yield different results, because the pre-training would actually be capturing some of the meaning of the genome. And this is actually shown, as you remember this figure I showed before, where the one-hot encodings actually outperformed these genome language model embeddings. In this paper, they suggested that stronger inductive biases towards learning relevant features in model architecture could improve genome LLMs. So essentially, if we could design pre-training tasks that enforced more biological priors in the pre-training, we could potentially have better downstream performance for these models. thank you so much for having me at this talk. It was a really great opportunity for me. I'd like to thank my supervisors, my co-authors, obviously all of the Harvard CCP team, my lab members, and the support that I've received from the Department of Computer Science. And special thank you to Alex Liu for the slide design.
Awesome. Thank you so much. That was a wonderful talk. I think, yeah, really appreciated just the breakdown and the overview of all of that stuff. So yeah, we have plenty of time for questions. I'm sure folks have a lot of burning questions, so I'll open it up. So feel free, yeah, just to unmute or post in the chat. I think Robert has a question.
Yeah, great talk. Thought it was quite interesting. To what extent do you know whether anybody's starting to use LD and how they, if so, how they're thinking about it? It's kind of hard to believe that you're going to get very good models without it.
You're saying like in terms of the pre-training task, like thinking about how tokens are related by linkage disequilibrium?
Yeah.
Yeah, I actually haven't heard anything about that. It could be that some people are secretly working on it to get ahead, but yeah, I'm not sure. It's definitely an interesting idea that it's like we're treating all of these tokens as independent and the masking task is independent, but many of these tokens are like strongly associated by evolution. So yeah, potentially it could be something that the model learns without being explicitly shown in these multi-species pre-training approaches, but I'm not sure anyone's shown that it can actually recapitulate LD.
Yeah. Okay, thanks. And then the second just comment is, mice are great, except we have no good outbred populations that get sequenced. So like it's you're better off working on almost any other species that was captured wild, because the mouse data is just going to be so bizarre.
Okay, well, I'll keep that in mind. Thank you.
Yeah, maybe I go next. Thanks so much, Michaela, for coming in this, I think, like really fantastic talk. I also found your preprint like really instructive to get up to speed in that topic. So thanks for that. I think you have started to talk a little bit about the limitations, and I've been very interested in, like this non-sequential data, single cell transcriptomes, and some of these tools like SCGPT and GeneFormer. And you might have seen this preprint who has basically said, like zero-shot performance of these tools are not very reliable, or at least not more reliable than simpler models such as SCVI or a simple, like highly variable genes clustering or integration. I was just curious, what is your take on this one? We have, I think, is also seen a similar like observation for like cell segmentation, where also simpler models were like outperforming like these transformers. Is this all like a question of having more data and more biological relevant data, or what are your thoughts there?
Yeah, I think that's a great question. I am familiar with the paper where they evaluated the zero-shot performance of GeneFormer and SCGPT. And I think, this shows that we have, a long way to go still in designing these genome LLMs. And I think part of that is, as I mentioned, due to the limitations of the pre-training that we do. We essentially just adapt directly from NLP and like kind of treat, like genomic sequence data, or even like in the case of single cell data, which is not even sequential, we kind of just like force it to fit this sequence idea and train on it with these pre-training tasks that have been shown to work for human language. But genomic information is very different, and there's an element of a lot of noise in these data sets that isn't present in the human language. So I definitely think there is a lot of work that can be done in improving the pre-training of these models to better capture meaningful biology. And I think also, the curation of the data is very important, as always. But really, I think it comes down to like these models that are being put out have really great performance, fine-tuned, and they're being proposed for all these fine-tuning tasks. And there's, a bit less work being put into evaluating them at the zero-shot level and showing that they can actually outperform these more simple models. And I think, it's very important to investigate the zero-shot performance of these models if they're being proposed as, foundation models, because, we don't want to be spending all of this compute on pre-training them if there's much simpler models that take much less compute to train that perform better. So yeah, I definitely think it's like, there's still some work to be done in that area. And these are kind of like the preliminary results of these models that are coming out, testing new things. They're on the frontier. So yeah.
Yeah, I had a question that was maybe related, similar to what Robert was talking about. Was so for, when you're talking about like these multi-species, like leveraging the multi-species data, like I wonder, have you seen any like research out there that looks at like the quality of like the pre-training like data? Like, because I know like even in non-model species and even in model species, like the genome quality like can vary so much. And so like do you know if there's like research out there that's like, here's the level we need it to be for it to be like accurate, or yeah, or like how much data?
Yeah, that's a great question. So I think there have been some, initial investigations into scaling, and it just shows, like of these genome language models specifically, and it just shows, like more data improves the performance, more data is better, but you also kind of need to scale the model up to be able to take in more data. And then once you scale the model up, you need more data, and like that's the trend. But I don't think there's been, at least not to my knowledge, there hasn't been a detailed breakdown of like, the quality of the data sets and like the differences in that. There's kind of just been this consensus now that, leveraging the multi-species has better performance. And, the question I proposed that was because of the evolutionary conservation between this data, which is what I believe, but it could also be like, it's just more genomes, it's just more data. And, that might like in some ways kind of address that, some of this data is worse quality than others. So you just kind of like put more data and hope for the best. But yeah, I don't think there's been a detailed analysis of kind of like quality and quantity of these genomic data sets. These are very much models that are being proposed a lot of the time by people with a strong computer science background and less of a strong genomic or like bioinformatics background to like look at these data sets. And the hybrid transformer models like that are trained on the genomic data, they have very carefully curated data sets because they're supervised, and like the quality is like very, it's up to a certain standard, and the evaluations are shared across these supervised models. And I think it's a bit more complicated for these genome language models. Like what is a good zero-shot evaluation to compare these models at, and like how do we measure whether the data set is good quality or not, and so on and so forth. So I think there's still a lot of open questions in this area.
Following up on that question, but I have another one afterward. You talk about evolutionary information presence in the sequence stuff. It's curious that you have this advancement in the protein sequence space, but not in the genomic sequence space right there. So the answer is simply that we don't have enough end products labels for where these things, because not the entire human genome is only a fraction of it is protein coding. Is what's the missing context for that kind of predictive power?
Yeah, so there definitely is a big gap between, for example, like protein language models and genome language models. Protein language models are performing much better. I think we can all agree. But I think part of that is like, as you were saying, in my opinion, I think part of that is like you were saying, the human genome, like a very small portion of it is coding, and the vast majority is this non-coding regions. And some of that we know may contribute to regulating the coding regions, but like a lot of it, we don't know what it's doing, or like it could not be doing something, or it could be, and there's no good way to kind of like balance the level of noise to not noise. Like we don't have a good way that we know how to do that. And like I propose the idea of the multi-species as like a way to kind of like, differentiate noise from not noise using evolution. But yeah, these data sets are very, unbalanced in that sense. And the protein language models have a much more balanced data set. The amino acids, all contribute meaning, and it's kind of a bit more related to natural language. But like protein also like exist in like structural space, and like they're missing an aspect of that just in the sequence data when they use that for modeling. And I think there's also a recent paper that's come out looking at the zero-shot capabilities of these protein language models and how they haven't quite yet reached the scaling law gold standard of more data, more performance.
Thank you. And for the other question, I'm not sure if I don't want to take more time here, but very quickly, how much pre-training do you think that we have to get to? Because I'm trying to figure out a lot of these downstream tasks, these task-specific context stuff. Is there any work on trying to more generalize them right there, transfer learning, task-specific context stuff? Because ultimately, you're just filling a mask for maybe whatever your genome sequence length, if it's length of 6 nucleotides, you're filling in the blanks each time right there. So I'm trying to struggle. Unlike NLP, we don't have a lot of good generalized downstream task which requires like human loop reinforcement training right there, which work well with NLP. I'm trying to figure out, is there any work on the downstream tasks? Because if people are just going, you can have a giant pre-trained stuff, but it's probably not going to be very useful for biologists unless you find a good task for them. And I'm struggling looking at Rudy's genomic stuff to find tasks that are generalizable, so that way it's good enough.
Yeah, so you're saying like beyond just like the pre-training, maybe like there are some general, like a more general like fine-tuning tasks that we could like fine-tune the model on, and then that could be used maybe for a variety of tasks. So kind of like a second stage of pre-training. Is that what you're talking about?
Something like that, but also like task-specific context attribution right there to identify relevant regions for specific predictions right there. Let's say you're looking for promoters or transcriptomes, but they have some further upstream stuff than from the downstream task. You would have that context right there, but I can't find any good ways of fine-tuning that, because fine-tuning just seems to have labeled data that goes through that. This is not necessarily, I won't have a lot of fine-tune. I just want to look up to upstream context, see if that's relevant or not. If it's yes or it's a yes or no binary classification, and that's kind of work doesn't seem to be done way well. It seems to be on a very specific lab by lab basis. I'm just wondering if every lab has to have a CS student to do that. I don't know how this is going to gain traction, because not every biology lab is going to hire CS students.
Yeah, I think that's a good, definitely a good point. I think there is an effort to kind of curate these data sets that like are actually representative of some of the tasks that biologists are like interested in, like not just like simple classification, but like these more complex data sets that can be used for downstream tasks like splice site prediction and other things. They're trying to compile this like benchmarking for all of these models so that they can be evaluated equally. And I think the question of what are like tasks that we can assign these models to better align their downstream tasks that they actually are going to perform on and be evaluated on is really important. Better alignment in a pre-training stage leads to better performance in the downstream task, obviously, because it's not learning information that isn't super relevant. The question of like how to train on a large amount of data that isn't necessarily labeled in a way that conveys good biological meaning that can work across a variety of downstream tasks is very much an open question.
Okay, thank you.
All right. Thank you so much again, Michaela. That was a wonderful talk. And actually, I'm going to hand it over to Sinclair just to give an update on the CCB seminar series.
Yeah, thank you. Thank you again. And just in the chat, on Monday at 3 p.m. in person, we do have another seminar with Glenn Coe from Stochastic AI. He's going to talk about his founding story and a little bit behind the tool he's developed. And on Tuesday at 5 p.m., we have Ariel Stern coming for Tannenbaum Healthcare Transformation Initiative. She'll be giving a talk on augmented reality in the operating room. It's pretty interesting tool that was developed and specifically with global applications in mind. So love to see you all there, and food will be available. So another reason to come. Have a great one. Thanks.
Thank you.
Thank you.